In week 1, we had a look at the basics of autophagy. To summarize what we covered in week 1, we discussed how cellular components are sequestered into membrane vesicles called orthogosomes and delivered to lysosomes in mammalian cells or the vacuole in yeast and plant cells for degradation. We also saw what autophagy actually looks like by looking at electron microscopy or fluorescence microscopy photographs and learned the fundamental roles of autophagy in our bodies, focusing on promotion of cellular component turnover, starvation response, and the elimination of harmful material from the cell's internal environment. This week, we'll have a look at the Nobel Prize-winning career of Dr. Osumi, during which he made a major contribution in understanding how autophagy works. Dr. Osumi's work on autophagy was conducted nearly entirely using yeast. So first, I'd like to talk about why we use yeast in life science research. The use of the budding yeast, Saccharomyces cerevisiae, has a long history in human culture and industry. Many of you will know that over many years we have used yeast to make all kinds of foods and drinks through its ability to ferment and brew. But we have also used yeast as an important tool to study biological phenomena occurring within cells. This is because the basic cellular activities we can observe in yeast are generally conserved in human cells, even though yeast is a single-cell organism. Scientists using yeast have discovered the mechanisms of cellular activities, for example, cell proliferation and the intercellular transport of materials. These findings promoted research in other organisms, including humans, and the Nobel Prize has been awarded many times to scientists using yeast in their research. Budding yeast is the most thoroughly studied, well-established model organism in the field of basic life science. This is because its genome sequence was the first to be determined among eukaryotes. It has a short generation time, which allows us to promptly progress research. And it is genetically tractable, which means that we can easily isolate its mutants defectively in a biological process of interest. These factors let us identify genes responsible for the process under investigation, as we are going to see in the case of autophagy this week. Autophagy therefore offers a key example of the power of yeast as a model organism in biological research. Autophagy was first discovered in mammalian cells by electron microscopy in the 1950s, and its key features were described by researchers in the US and Europe. However, the physiological role and mechanism of autophagy remained unknown for a long time, because researchers could not find the genes that controlled the autophagy process. If we know what genes are required for autophagy, we can manipulate or disrupt these genes and make cells in which autophagy doesn't happen, letting us find out what role autophagy plays in the cell. On the other hand, if our objective is to elucidate the mechanism by which cells undergo autophagy, we need to analyze the functions of proteins, which are the products of the responsible genes. Therefore, at the time of Dr. Osumi's work, the identification of genes involved in autophagy was essential for the development of autophagy research. Dr. Osumi's major contribution was to not only discover that autophagy occurs in yeast, but also to identify virtually all of the autophagy genes. This triggered the explosive development of autophagy research in many labs around the world in a variety of organisms. Following close behind Dr. Osumi, several other groups in the US and Europe also find genes involved in autophagy using yeast species, which partly overlap with the genes Dr. Osumi identified, but here we will focus on the story of Dr. Osumi's discovery of autophagy and autophagy-related genes in budding yeast. Before starting his studies in the field of autophagy, Dr. Osumi started out as a researcher studying the function of the vacuole in yeast, and he demonstrated that the vacuole is a much more active organelle than researchers had assumed at the time. But once he got an independent position in the University of Tokyo, he decided to set out in a completely new research direction. In those days, people knew that the vacuole contains a number of degradation enzymes, but didn't know how cellular components are delivered into the vacuole for degradation. This puzzle interested him. It was known that yeast cells are converted to a special type of cell called a spore when they are shifted to starvation conditions. The formation of a spore is a massive change for a cell and requires a vast remodeling of the cellular components. As cells subjected to starvation don't have free access to external nutrients, Dr. Osumi assumed that during such a big change, not only synthesis but also large-scale degradation and recycling by the vacuole would also be important. He also knew that the vacuole is an organelle which can be observed under a simple light microscope, as shown here with red arrowheads. Note that these cells were grown under a nutrient-rich condition and the vacuoles appear to be empty. He hypothesized that cellular material delivered to the vacuole could be observed under a microscope if degradation enzymes within the vacuole are inactivated. What he tried was very simple, but no one had done it before. His simple but ingenious idea was to observe yeast cells deficient for vacuolar degradation under starvation conditions. Using this approach, he observed many particles vigorously dancing about within the vacuole, as you can see in this movie. A close investigation by electron microscopy and biochemical analysis revealed that these particles contain cytoplasmic components and allowed Dr. Osumi's group to conclude that a phenomenon known as autophagy in mammals was also induced by nutrient starvation in yeast cells. In this electron microscopic image, you can see various vacuoles marked with blue circles, which contain many autophagic bodies corresponding to this structure, which is released into the vacuole after fusion of the autophagosome with the vacuole. As I mentioned, one of the biggest advantages in using yeast in life science research is its genetic tractability. Immediately after the discovery of autophagy in yeast, Dr. Osumi's group proceeded to aggressively pursue the isolation of mutants defective in autophagy. They treated yeast cells with a chemical that induces mutations at random positions in the cell's genomic DNA, and using a microscope, looked for mutants that do not accumulate autophagic bodies, even after shifting to starvation medium. After observing no less than 5,000 mutants one by one, they finally succeeded in obtaining a single mutant which did not accumulate autophagic bodies within the vacuole. As you can see here, in contrast to parental cells which accumulated autophagic bodies, no autophagic bodies at all were observed in these mutant cells. Following a convention for naming genes in yeast, they named the mutant APG1-1, where APG stands for autophagy. In this mutant, a certain unknown gene required for autophagy was being impaired by a mutation. Dr. Osumi was not satisfied by this achievement because he assumed that autophagy, which is a highly dynamic and complex phenomenon, must require many more genes. However, since only a single mutant was obtained after the examination of 5,000 mutants, they decided that continuing the same approach was not the most efficient way forward. In the meantime, they made an important discovery. The APG1-1 mutant, which is defective in autophagy, died earlier than normal cells under starvation conditions. They decided to take advantage of this phenotype for the isolation of more autophagy mutants. In their second attempt to obtain autophagy defective mutants, they started with about 38,000 mutants and first picked up about 2,700 mutants sensitive to starvation, which could be easily found on agar plates using a dye which stains the dead cells. Next, they examined mutants individually by microscopy to check for the accumulation of autophagic bodies. In this second step, the examination of 2,700 mutants resulted in the isolation of 99 autophagy defective mutants. This is in contrast to the first study, in which a single mutant was obtained after examining 5,000 mutants, so the efficiency of mutant isolation was greatly improved by adding a simple step. This is an awesome example of genetic screening for mutants and the power of yeast as a model organism in biology research. Our further genetic analysis of the 99 autophagy defective mutants uncovered 14 genes that were mutated, causing the observed defects in autophagy. Here is a list of the 14 APG genes Dr. Osumi identified. Later on, these genes were renamed ATG, which stands for autophagy related, to avoid confusion, because several other groups identified some of these genes later on using different approaches and reported their findings using different names. Identification of the autophagy genes in yeast and the determination of their sequences immediately allowed us to discover equivalent genes in other organisms, including mammals, just by searching sequence databases on a computer. As I mentioned earlier, the inability to identify the genes controlling autophagy was holding the field back, and this work allowed researchers all over the world to investigate all stages of the autophagy process. Indeed, researchers around the world began to create autophagy deficient cells or organisms in line with their interests and investigate the implications of autophagy disruption. Collectively, this work revealed that autophagy is involved in a really wide range of biological phenomena beyond anyone's expectations, and also demonstrated links to a number of diseases in humans. We will talk about this in further detail next week. Further progress resulting from the discovery of the ATG genes was the identification of a marker to label autophagosms, which gave researchers an important tool in autophagy research. A protein named ATG8 in yeast, or LC3 in mammals, which is encoded by one of the ATG genes, was found to exist on forming and complete autophagosomal membranes. By expressing these proteins as a fusion protein with green fluorescent protein, which is usually called GFP, researchers were able to observe autophagy in living cells under a fluorescence microscope. As you can see in this movie of living mouse cells, shifting cells to starvation medium results in the formation of many puncta of GFP-LC3, representing autophagosms. In this way, researchers could see the induction of autophagy in a much easier way, in real time and using living cells, which was a major improvement in fluorescence microscopy. Moreover, transgenic mice expressing GFP-LC3 were generated in the lab, which allowed us to know when, where, and how much autophagy is activated in a whole mouse body. The molecular mechanisms of autophagy have also been thoroughly studied by examining the ATG proteins. ATG proteins form several functional units, including complexes for protein modification and lipid modification, a membrane protein localized to a small vesicle, and protein conjugation systems. It was subsequently found that these proteins get together at the site for autophagosome formation and cooperatively act to form the autophagosome. In particular, recent studies greatly extended our understanding of how starvation signals are transmitted to particular ATG proteins to induce autophagy. This information about the mechanism of autophagy also provided us with an important basis for understanding the physiological function of autophagy in our cells. This graph showed the number of research papers published in the field of autophagy over the last 30 years, which is an indicator of research activity and general interest in the field of autophagy. As you can see, the discovery of autophagy in yeast, the identification of ATG genes, and subsequent elucidation of the basic mechanism of autophagy triggered the explosive growth of the autophagy research that continues today. This culminated in the award of the Nobel Prize in Physiology or Medicine to Dr. Osumi in 2016. Before we finish this week's lecture, I'd like to add a couple of points about his achievements in the development of autophagy research. Dr. Osumi not only made pioneering discoveries using yeast, but also greatly contributed to the development of the autophagy field, allowing research to progress to the next stage. Based on the information obtained by analysis using yeast, he immediately initiated the study of autophagy in mammals and plants in his own lab. He also used a whole range of techniques, including genetic, morphological, molecular biological, and biochemical approaches, and initiated collaborative research with structural biologists to determine the structures of ATG proteins. This promoted a comprehensive understanding of the mechanisms and roles of autophagy. In this way, his group has produced an essential body of many outstanding results, and while the tremendous achievements of many other researchers around the world were important in the development of autophagy research, Dr. Osumi's group played the leading role in the field. Finally, I'd like to emphasize that Dr. Osumi never set out to receive the Nobel Prize or to directly find a cure for any particular disease when he began his research in autophagy. His study has always been driven by his pure curiosity and desire to learn about the basic function of cells.